Each system, that means if this cell is a part of a respiratory system, so the cell of this system perform the whole function of this system. So the cell make the same system, function of this system. So cell is a building function unit of systems. Okay, now I want you to ask yourself about the size of systems, size of cells are same or different. Cells different in many types, in many ways. First one, different in size. So cells different in one size. Number two, structures. Number three, functions. So cells different between us in three rows or three branches: size, structures, and function. Give me an example, okay? Your examples about different cells. Like first one, red blood cells. Red blood cells, part of the blood that is responsible for carrying gases like oxygen, carbon dioxide, and also carrying food like digested food and wastes. So red blood cells like its shape, okay? It's concave and nucleated, no nucleus, okay? Empty from nucleus. Second one, okay, nervous cell. Nervous cell or neuron by its shape, body and extended from the body, dendrites like this shape, okay, and has nucleus. Neuron or nervous cell may be its size different from microscopic or be very long cell, maybe 1 neuron extended from 1 meter to more, to link between brain, spinal cord, and the smallest joint in your neck. So the size is different. Okay, number three, give me another example, like muscular cell. Muscular cell is the building unit of a muscular system. Muscular cell, named also by muscular fiber, by this shape, this like slender shape, and striated, constricted, okay, right, and multinucleated, okay, contains a lot of nucleus. So its work about contraction and relaxation is like the motor instrument inside your body to contract and relax. If my arm contract, I can elevate this shape up by relaxation, so it will get down. So it's like a mechanic inside your body. Okay, number four, the largest, the biggest one in the life size cell is the egg, but not all the eggs are fertilized eggs. Okay, and the biggest one, like the egg of ostrich. Fertilized egg meaning ovum without sperm, without fertilization. So here, as you see, the size, okay, different. The biggest one, egg of ostrich, the biggest cell. The smallest cell, bacterial cell. Bacterial cell. It is the smallest cell ever. Okay, consists of only cell, and it's from prokaryotic, meaning of prokaryotic, no living organism. Okay, living organism, no nucleus inside, and consists of only membrane from outside and cytoplasm and classified into two types. Living organisms, okay, classified into unicellular or multicellular. Meaning of unicellular, the whole body of the living organism consists of only one cell, while multicellular consists of multi or many number of cells. Give me an example on unicellular. We have a lot of examples, but all of those microscopic, meaning very tiny in their size, like bacteria, paramecium, amoeba. Okay, and we have also from fungi like yeast, fungus. All of those are unicellular living organisms. While the other side, multicellular. Multicellular consists of many of cells, like many of the living organisms, like human, like plants, like most of fungi, like algae, and so on. So we have here diversity in also like living organisms, diversity in the types of living organisms, diversity also in types of cells. Now, how can I know these structures? By aid of some scientists. Some scientists made and performed a lot of theories. First one, and have a lot of effort to prove the presence of cell. Number one, Robert Hooke. Robert Hooke, the first scientist that invented the simple microscope, but not that like this shape, okay? Simple microscope consists of only one lens at each side, like this shape. Okay? By this shape. And can observe what? Observe a part of plant. This part is named by cork. After observation, found that cork consists of rooms and name these rooms by cells. Cells, this name back to the Latin name, okay? This Latin name is cellula. So cellula is the Latin name that derived cells from it, okay? By the effort of Robert Hooke. Robert Hooke, the first scientist who described and observed cell, okay? Number two, the first scientist that described microscope and invented one. Number two, second scientist second scientist, about 9 years difference between Robert Hooke, okay, worked on world in examination and also made another microscope. This microscope has more magnification power, about 200x times of Robert Hooke microscope. Observe words, it's very important words, okay? Observe words like blood, okay, water, bone. By observation of water, bone can describe the microscopic living organisms. Okay, so it's a measure of the volume. Number 3 and number 4, complete measures. Okay, number 3, skeleton. This also scientist, okay, German scientist. It's worked by description that all plants consist of cells. So it's effort that all plants consist of cells. The last one, sorry, number 4, okay, Schwann Rudolf Virgil. Rudolf Virgil, okay, he made the basics of the cell theory about, first one, each cell had to come from pre-existing cells. So we cannot get cell from parents. So each cell gets from pre-existing cell. Number two, okay, that cell is a building and function unit. These 2 points from 3 of the cell theory. Now we are going to know the cell theory. Cell theory about 3 points, 1 and 2. Each cell gets from pre-existing cells. Number 2, cell is building and function unit. The last one, number 3. Number 3, that living organism may be consist of one cell or multi-cell. So you need or multi-cell. Okay, this is the cell theory and the assumption of some scientists. Okay, now if I want to describe and study the cell web, so I have to this help is a microscope. This type of microscope, I think you saw before inside your schools, okay, named by light microscope. But we have two main types of microscopes. First type of microscope, light microscope, and the second one of microscopes is an electron microscope. So we have here 2 types of microscopes. First one, light microscope. Number 2, electron microscope. Light microscope, okay? Light microscope used by scientists till 1950, okay? 1950. Electron microscope used after this date, 1950. So till this day, we didn't know any microscope except the light microscope. Light microscope it consists of its types. We have 2 types of light microscope: sample microscope and the compound microscope. Meaning of sample and light microscope. Focus field, okay? Light microscope consists of main parts, okay? First part, this part is lens. Its type is convex lens. Convex lens to what? To get light and to enlarge the size of the spacement or the sample, okay? This means of convex lens. This convex lens, where I see the sample from it, I can name it by ocular lens. So the first lens is an ocular lens. This part is rotating part, okay? Consists of group also of convex lenses named by objective lenses. So we have here 2 types of lenses: ocular lens and objective lens. Any microscope consists of also some other types that share in clearing of the sample, okay? This is the sliding bar, and we have here stage. This stage, I can put the slide here, okay? And this is a light microscope. How can I enter the light? By this mirror. This mirror from type concave mirror that collect light and concentrated into another part here. This part is named by condenser. This condenser collects the light and reflects it into the sample here that pass light to the lens, then transfer it to the ocular lens so I can see the sample. How can I control the space of this sliding part? Okay, by two adjustments. We have here two adjustments. Okay, the upper one is a coarse adjustment, while the lower one is a fundamental adjustment or fine adjustment. Okay, this is the complete structure of a light microscope. This type is compound light microscope. Why compound? Because it consists of one or two ocular and multi-number of objective lenses, while the second one consists of one ocular and one objective. So the random one is the compound light microscope. The main idea of a light microscope is using light. So the main idea here is by using light. Can the light pass and I can see the sample? Sample here will be more than its real size. The magnification power of this microscope can reach till 1500 the real size of the sample. So, magnification power here, okay, about 1500x. How can I calculate the magnification power of light microscope? By multiplying ocular lens in the objective lens. So this is the total magnification power of the light microscope. Magnification power equal power of ocular lens times power of objective lens. So this total have to be more reach to 1500. If the total magnification power increased more 1500, this will be very wrong. Why? Because after 1500, the image will be uncleared. Uncleared. Okay, this is very important. So you must know when you solve this type of problem, the amount of total magnification power cannot increase more than 1500x. Okay, this is type of light microscope. How can I increase the clarity of the sample? By two ways. First one, by putting the sample under the light microscope. First one, dyeing it. Meaning of dyeing, staining. Staining, it changes the color of the sample so I can see it clear. Or number two, I can change the color of the light. Light consists of 7 spectral colors as you know before. So by difference in those spectral colors, I can see each part inside the cell well. So we have here two ways. Which one the preferred one? Not here preferred as we know where I work on. If I worked or observe a unicellular cell, you cannot here use dyeing or staining. Why? Stain here can kill this cell because it's unicellular, so can affect it hardly. So this is the first type light microscope. Second type electron microscope. Electron microscope. We have here two types of electron microscope. Before knowing the types of electron and more from the real size. Okay, can you imagine? I can increase the size of a small, very small thing, million parts and more. It's very huge. So now I want to know the types of electron microscope. We have two types of electron microscope. First one, tell me about the structure of the surface of the cell. So the first type, tell me the structure of the surface web. Second one, tell me the structure of the parts inside the cells. So we have here two parts. Electron microscope types. Number one, scaling electron microscope. Number two, transmission electron microscope. First one is scaling light electron microscope. This type shows the surface details like white blood cells. By scaling, I can see its surface by this shape. This is about the amount of the magnification power here, 3500x. Transmission meaning showing the details of the cells. Meaning of details equal organelles. I can study by this type of microscope the details or the organelles inside. Okay, so the structure of the white blood cell. By this shape, I can see here the shape of the nucleus and its structure. Okay, this is magnification power about 8900x. Thank you, and I want you all wishes in